Welcome to ASM Live at ICAAC. I'm Michael Schmidt. I'm one of the moderators of This Week in Microbiology, and I have the great pleasure of being able to visit with you here at ICAAC at the 53rd Annual Conference on the Interscience Conference on Antimicrobial Agents and Chemotherapy. Today we have with us Dr. Cliff McDonald from the Centers of Disease Control and Prevention, and Dr. Alan Walker from the Sanger Institute over in the United Kingdom. And they're going to share with us the exciting developments that they're going to report on a session here later this afternoon on the role of the microbiome and infection control, specifically how antimicrobials are interacting with this emerging field. So we're going to start with Cliff. Cliff has the delight of having been one of the co-organizers of this session, and he's going to share with us the vision of the organizers of this very provocative session, and then we'll have Alan share with us some of his exciting results that he's going to share with the audience later today. So Cliff?
Yes, the microbiome has been capturing the attention of all of us in recent years, mostly because of the revolutions really in genomics and metagenomics and the ability that affords us to enumerate and understand the very complex nature of the microbiome. And the context, most of the context of the discussion today will be the lower intestinal microbiome, but of course it's not limited to that, but certainly the most complex. Only 20% of the organisms in the human lower intestine have ever been cultured successfully in vitro. And sort of as I always give the analogy, it's like the Mariana Trench of unknown.
Well, they're mostly anaerobes, and that presents a challenge unto themselves, because how do you get them out in a living condition?
Right. So right now, because of metagenomics, we can get an understanding and get a look into it. And what we're realizing, of course, is that every time someone takes an antibiotic, or even other drugs too, it's not just limited to antibiotics, and of course it even extends to diet, but within healthcare, it's antibiotics predominantly. Profound perturbations occur. And one thing that's brought this to the head has been the epidemic of C. difficile as well. And we've long known that antibiotics were so important for predisposing to that. But we really, as the organizers, really want to go even beyond that, use what we're learning in C. difficile to understand that C. difficile is one of several multidrug-resistant organisms. Organisms or strains of species or strains of species that have a selective advantage when antibiotics have perturbed the microbiome. And C. difficile really optimizes that, and it's part of its whole pathogenicity. But several other multidrug-resistant organisms, whether it be vancomycin-resistant enterococci, or now carbapenem-resistant enterobacteriaceae, or the extended spectrum beta-lactamase-producing enterobacteriaceae, all of them colonize persons either in the lower intestine or on some other site of a biofilm, and that's another area we need to address. But just in the area of the microbiome, the lower intestine, this is where they colonize, and when they colonize the patient who's had antibiotics and has this perturbed microbiome, they don't just colonize, they multiply, they expand.
And then you move them in the environment of the hospital.
Exactly. But it becomes very hard to not move them when the gradient, if you want to say, gets so great that they expand greatly in the person who's had a perturbed microbiome, and they become contagious. Those persons become contagious.
And the principal readout for a C. diff perturbation is diarrhea. Right. And that's extremely difficult to contain.
Exactly, all the more difficult.
And it's further compounded by the fact that C. diff makes spores.
Right.
So even though they're God-fearing anaerobes, as they say, that are killed by molecular oxygen, the fact that it makes a resistant endospore just enables this microbe to spread so much more easily in the environment of the hospital, and it's literally moved on the winds.
Right. And the early event, though, was the exposure to antibiotics that made that all possible. That was the trigger. And so we're not going to get rid of antibiotics. They're here to stay. They have saved countless lives. They are a precious national and international resource. And when I say a resource, protecting the activity of them is a national and international priority, public health priority. Well, the main emphasis in public health has been, well, twofold. One is to prevent cross-transmission. But as you just mentioned, once these organisms have expanded in numbers, grown up in numbers, trying to prevent transmission can be very difficult. Not that we shouldn't do more, and we will continue to do all we can. With the problem with carbapenem-resistant enterobacteriaceae, we are not, right now in the United States, still very much in a containment mode.
We're tremendously lucky. Our friends in the United Kingdom aren't as lucky as us.
With regard to that resistance, exactly.
With regard to that resistance in Northern Europe. And it's really quite frightening how quickly that microbe moved from India to the Northern Europe and then over to the UK.
And I'm afraid it is moving here in the United States.
Oh, no,
and
it's coming.
We don't know it.
It's coming.
And it sort of comes out of the woodwork because it does spread from person to person. And you mentioned diarrhea and C. difficile. We know that at least. At least you can identify a patient. But when someone's heavily colonized with CRE, we don't know it unless
until
you look. Until you look.
And that's a very expensive proposition in the prophylactic state, where you're asking the hospitals to do the surveillance on their own nickel, because they don't necessarily get reimbursed.
So we have to buy time with transmission interruption, and we will continue to do that. But the long-term solution is going to be to manage the microbiome. I like to say tending the microbiome. Thinking of it as a garden.
Yes.
And it really is. And that's one way to think of it, and often I talk about it that way. If you spray Roundup or a weed killer on your garden, what will grow up but invasive species? And that's exactly what happens here in our patients over and over again. We are not going to do away with these antibiotics, so we're going to continue to lean heavily upon them. Every time we're doing aggressive therapies, surgeries, all types of heroic efforts to save people's lives, modern medicine is miraculous.
We couldn't do transplants
without... We couldn't do it without antibiotics. So what must we do? We need to use these more in the sense of tending the microbiome. Realize we will disrupt the microbiome when we give this antibiotic. What can we do to restore that microbiome? What can we do to bring back the bacteria that are not the invasive species, not the multidrug-resistant organism? Or what can we do more smartly with antibiotics? Narrower spectrum antibiotics. Is there a place for antibiotics to be developed that maybe only get one species? Now realize that's a hard hill to climb right now, because how do you use those antibiotics empirically? You can't. You only can use them after you have a diagnosis. There's a lot of challenges in that, in terms of a market, in terms of a whole paradigm for that.
Well, how do you even afford to develop a narrow spectrum, single organism antimicrobial?
Challenging. Okay, well that's one hope, though, still, that some like that could be developed in certain niches. Another would be some type of agents that might bind up antibiotics. So while the antibiotics are being used, that they might do something to inactivate the antibiotic in the microbiome, or somehow otherwise prevent the damage.
Sort of beta-lactamase on a different scale.
That's a different thought, too. Yeah, and it's deactivating. And then going down the pathway, of course, shortening the antibiotic courses, we're doing those things. Maybe even gauging patients' risk. And that's something I'll talk about, that the idea in 2020 that we may routinely, or some years out, maybe routinely, as patients leave the hospital or after they've finished their antibiotics, check their microbiome.
See what they got.
Effectively
check their oil.
Right, and then if we have to give them back some microbiome that is more natural. Or another thought is even to bank their own microbiome before they begin a transplant procedure or long chemotherapy or something of this nature.
Well, I know the FDA is having active discussions right now of what is a proper donor for a stool transplant.
Right.
They're looking at those questions, because for the treatment of C. diff, stool transplants have become, since the New England Journal has published that seminal study this past January, it's of course become standard of care. They're really beginning to take it very seriously, and the FDA is looking at what is a donor.
Yeah. Let me say one more thing too, as you brought that up, it reminded me also, is that this issue of microbiome disruption, we're talking primarily about it being infection control and trying to manage it so that it doesn't spread to the next patient. But also, it may be a risk to the patient themselves. That's true in C. difficile.
Yeah.
But it may be true with some other organisms as well. Some work from Eric Paymore's group at Sloan Kettering suggests these are patients who are stem cell transplants. They're patients who are getting a lot of antibiotics, peripheral stem cell transplants for leukemias and things like this. They actually showing evidence that they precede into developing bacteremias with VRE and multidrug-resistant or just gram-negative organisms. There's a dominance in their microbiome with those organisms first. So that what seems to be happening in those very debilitated patients, at least, is the microbiome vastly disrupted, opening up this ecological niche. These weeds, if you want to say, are getting in there, growing up to high numbers, and then leading to bloodstream infections in these patients. Exactly how that all happens, we don't know, but just the risk of having a dominance of a virulent species, it may be a patient risk for an individual.
And it spirals out of control because once you get a bloodstream infection, the standard of care
then
is
antibiotics
an
again,
antimicrobial.
and then still very disrupted microbiomes. And that's the modus operandi now. I call it almost the scorched microbial earth, which is how we're practicing medicine now. We need to change the paradigm. We need to change the mindset. I want to say one thing here, and I think that especially for ASM and the field at large, that we need to get the sense that, at least from a public health standpoint, the most important event in antibiotic resistance is not necessarily the de novo genetic rearrangement or mutation. That's very interesting, and that's been a focus, and it's partly because it's very accessible to us. You take one organism out, you study in the laboratory. But from a public health standpoint, once that happens, it's probably more important how it spreads through the human population or through animal populations through the microbiome disruption. And that's where the antibiotic, and I say antibiotic pressure isn't antibiotic pressure, it's really pressure on the microbiome. And we should start talking about this. Antibiotic pressure on the microbiome, that's the most important for the spread of antibiotic resistance.
So we need to change the conversation from antibiotic stewardship per se, which is effectively just counting the number of scripts that are being written in the facility, to really understanding the microbiome of the community that are our hospitalized patients.
Right. Tending the microbiome. And you have actually brought up the fact that it isn't just in humans. It can get in the innate environment also. That can involve biofilms, which is another part of this conversation, which you could spend time there also. But also then there's a microbiome in food-producing animals and animals in general.
Which we use, I mean, it's all about farm to table.
Exactly. And the disrupted microbiomes leading to clonal expansion, which is happening over and over and over again. So I think it's a conversation for infection control. It's a conversation to help. How do we get the public to understand this? This is going to be a challenge.
So your session today is really a very narrowly focused topic.
Infection control.
Trying to look at it from the perspective of infection control, which is an extreme problem in the developed world and in the developing world. Because the microbes, it goes back to, I like your phrase, tending the microbiome, because we really have to pay attention what's going on, because that's the only way we're going to get a handle on infection control. It's almost, we can go to one of these new OM words, it's the envirome. What is the healthy envirome of a hospital?
Yeah, that's true.
I mean, do we even know that to a certainty? Because everyone is out there since the time of Florence Nightingale scrubbing the hospital clean. And as Phil Carling has so eloquently showed, is the United States spends on average about $10 billion a year cleaning our hospitals. And Carling and his colleagues with their magic glow dots have shown the busier you are, the less likely those objects are properly cleaned. And is cleaning. I saw a poster yesterday that the Acinetobacter are becoming resistant to traditional disinfectants used in cleaning hospitals. And is that, if you will, an antimicrobial that is disrupting the natural microbiome of the built environment that then will jump into the disturbance of our patients. And as you well know better than most, being at the CDC, Acinetobacter is a terror.
Yes.
It's a terror.
Well, within the patient, there's also this issue of biofilms. And even those, we did some recent work on needleless connectors to central venous catheters with metagenomics. You typically think of these biofilms as having maybe a few organisms, maybe a staph and a Pseudomonas or something, but they're very rich. They're very rich. When you look metagenomically, you see there's many more organisms there. It may be even that biofilms we can tolerate biofilms better than we thought if we start to understand the population dynamics of those. You're bringing up also the innate environment. A lot of the dry surfaces probably don't have as rich microbiome in general. The wet environments certainly do.
Oh, God.
And those are biofilms with communities again. So yeah, I think you're absolutely right. I think a lot of the hospital environment is very influenced by the patient microbiomes coming in and what we're doing to the patient. If we tend those patient microbiomes, we'll probably have better environmental microbiomes largely also. But your point is a valid one too. When you bring in disinfectants and things like that, can you open up niches out there also? And I think probably especially in the wet environment.
Well, we know that the microbes love to exchange information.
Yes.
And it's these ancillary environments that could be the, if you will, the genetic reservoir that can facilitate the transference of these antimicrobial markers into the patient when they're in a perturbed state, because perturbation, the Chinese have this phrase, chaos breeds opportunity. And this is, of course, certainly the case in the microbial world, is when you have that perturbation, they're more susceptible to conjugation, transformation, and even transduction. All the three methods of genetic exchange, and the microbiome is acquiring new data constantly. And we witness it. So what are we going to learn in your session? Take us through the hot topics that you have before us.
I mentioned some of them already. Eric Paymore's work, for example, just intro from some of Relman's work on the ciprofloxacin courses.
Dave Relman from Stanford.
Yeah, right. But also just sort of putting in this whole context of what we just discussed. It's really, you've heard much of it right now in terms of a capsule and just going into showing some of the data behind this. Yeah. And also I will mention one more thing, and that is what are some concrete steps that we're taking in the initial. Yeah, I think one of them is this idea of coming up with a microbiome or antibiotic disruption index. This is still conceptual right now, but a standardized set of how we might measure the disruption, that we could produce some standardized methods that people would start replicating. I mean, one thing we do at the CDC is we come up with a surveillance definition or a standard lab protocol or something else that other people will take off and start doing. We may be still early in this, but the idea would be to really perhaps FDA could even take it up. And we're working with FDA a little bit and dialoguing about this, that it could even become part of a pre-approval process for a new antibiotic, to understand what does that antibiotic do to the microbiome. How does it perturb? What's the standardized picture of it? In animals or in humans, in early studies, you could do it in humans as well.
Well, the revolution in MALDI-TOF is making it fairly inexpensive to look at many of these things. Since it's proteomic, we can actually look for the gene products that are descriptive of antimicrobial resistance. And, MALDI-TOF is fairly inexpensive, and if you could develop a genetic profile from, a stool sample or something that is easily harvested, so you could, on discharge, you could acquire from the patient what their perturbed biofilm looks like, and then ultimately, once they get back to stability, have a reference, if you will, a reference stable fingerprint. So you could compare and contrast to see about these perturbations, because I think that's what our colleagues at the FDA are going to really begin to throw the new pharmacodynamic data against, because how badly does it perturb
Right.
in...
I think we're going to have to tie these to outcomes too, of
Yeah.
course. You know, and so what is the perturbations that lead to a certain risk for C. diff? What are the perturbations that lead to a certain risk for MDRO colonization or dominance with infection? I didn't even mention pathobionts. Come to my presentation, I'll mention that there.
He teases. He teases us. Well, this takes us to Dr. Walker's presentation, and of course, Clostridium difficile is a tremendously horrific infection. There are people who acquire this infection and then don't have foreign bowel movements for greater than 2-3 years. And it's really quite debilitating, and it would be really terrific if we could figure out a way to set them right. And that's, of course, what your magic has come up with. You know, fixing or restoring the garden to its original, pristine, perfect condition. And Dr. Walker here has developed a magic elixir to the people who have C. diff that hopefully will actually help you then learn how to, whether or not your garden tending is actually working. So Dr. Walker, tell us a little bit about your story.
Okay, yeah, thanks. And so, I mean, both of you have touched on the fecal transplantation aspects. And that New England Journal article is a perfect example of how to do it properly. And we now have really good evidence that this is a really, really effective last-line treatment. And actually, maybe we start thinking less of the last-line treatment and maybe more of a routine treatment. Of course, there are some major barriers to using it. It's not very pleasant. It's still feces, after all.
And getting the donor.
Well, I mean, this is one of the major issues. I mean, the big concern is infection control. Are you passing new pathogens from the donor to the recipient?
Yeah.
Right? So obviously, when you do this in the clinic, there's a screening program has to go on to make sure you're not introducing novel pathogens. As you said, so we've only cultured 20% of the microbiota. We don't know what the rest of them do, really. So even if you've ruled out known pathogens, there's still a theoretical risk that you're introducing something worse. So really, our interest in this issue stems from the fact that we knew fecal transplants worked, but how could we refine those? And so this is work really driven at our end by a guy called Trevor Lawley, who's a faculty member who developed a mouse model of Clostridium difficile infection and transmission. And really, we found the same issue you get in humans, where you get a proportion where you just can't shake off the C. difficile. Certain strains seem to stick. The patient ends up dependent on vancomycin, for example, and as soon as you take that away, you get relapsing disease again. And so it's a problem, like you said, where you get patients with 2-3 years stuck in a bed, they can't function, they don't have solid stool movements, and it really impacts greatly on their standard life. So for us, the issue was then, and for Trevor, the issue really is, well, what can we do in the mouse model? Can we identify novel mixtures which will work just as effectively as whole fecal transplants? So what we did is we took our mouse model of transmission and infection, did what they do in the clinic, gave fecal transplants. They work fantastically well. The mice are cured. C. difficile is wiped out from the system. It blocks transmission, really cures the problem. And so using that model, we then embarked on a sort of program of culturing to culture different strains. And this is one of the things I should really mention is that although we've only cultured 20%, and they are difficult to culture because they're strict anaerobes, they're not really unculturable, so to speak. I mean, it's just, there's not that much work done in this area. I mean, if you do it right, I mean, these aren't things that live in the soil and multiply every 3 months, right? They have to multiply quick or they're gone. They're going to get flushed out of the system.
Which is good news.
Which is good news, exactly, yeah. So, and you kind of know exactly what sort of strubbits they're going to be feeding on because it's the sort of things that are going to be present in the gut, right? So,
right.
So these things
are what we eat.
Exactly. So these things are definitely, to a large extent, we think, culturable. But there are only a few groups around the world really who are doing the sort of dedicated culture programs that involve specialist media, for example, that you need to grow these sort of bugs. So we'd established anyway that fecal transplants worked fine in the mouse, they cured disease. So I mean, the rationale was, right, let's simplify this. So we tried sort of passaging the feces overnight in bacterial broth to simplify the sort of things that were growing. We went through serial passage, and the mixture got simpler and simpler and simpler, basically enriching for things that were growing in the media.
Classic enrichment techniques that Stanier taught all microbiologists from time immemorial.
Exactly. It's not a new thing. It's really just applying old ideas to these sort of new areas of interest, which is infection control with the microbiota. And lo and behold, it worked fantastic. The first step was really we had a mixture of 30 species that worked just as effectively as whole fecal transplants. Yep, worked just as well. The mice were cured, transmission was blocked. So then we asked the question, just how simple can we make this? And so we tried individual strains, and none of them worked on their own. So we think really the key is that you need some form of diversity there. And that fits on what you're saying. It's tending your microbiota. It's restoring the microbiota diversity. So, but again, the more diverse the sample is, the more difficult it is to make into a therapeutic, right? So there's a balance to be sort of gained there. So I think the best we managed to do was to get it down to 6 strains. And this was a mixture of strict anaerobes, and 3 strict anaerobes, novel species, never cultured before, and 3 facultative anaerobes. So you have a mixture of the 2 different sort of growth behaviors. When administered to the mice, worked just as well as all fecal transplants. Beautiful restoration of the microbiota. What we think happens is that these 6 strains essentially tip the balance against C. difficile and some of the co-occurring nasties you talked about, these weeds that grow in at the same time. And that seems to have tipped the balance in favor of some of the suppressed, healthy microbiota, which was there, but at very low levels and was being kept sort of under check.
So you allow them to effectively come back.
Exactly, yeah. And so what happens is that the C. difficile drops a little bit, and these other co-infecting microbes drop, and you get this bloom again in the healthy microbiota. It regrows, and essentially once it's established, you've got colonization resistance infection again, which means C. difficile is kept in check. The mice are no longer infectious, the disease is cured. So it's really, I think, a proof of concept. It's only a mouse model, of course, but it's a proof of concept that you can recapitulate the same effect you get with fecal transplants with a small number of cultured strains. So I think that's really exciting. I think it opens up all sorts of possibilities, not only in C. difficile research, and there will be a number of papers coming out over the last year or so, I guess. There's a proof of concept out demonstrating that culture strains work in C. difficile. This was out early this year. There's some work just came out in Nature showing that 17 Clostridia strains from the gut can induce regulatory T cells, and so therefore have an anti-inflammatory effect in cases of allergies and colitis. And then finally, just last week, there was a paper out in Science showing that cultured organisms from lean individuals can protect mice from weight gain. So there's all sorts of potential.
So obesity, you've solved obesity.
Not me, you have to say.
No, but this is absolutely where understanding this whole tending to the microbiome using a very defined and focused area of infection control where we have easy outcomes to measure.
Short-term outcomes, right.
Short-term outcomes where we'll develop the necessary methodology to go after the holy grail of obesity in type 2 diabetes and chronic inflammation and Crohn's disease and all of these other things that we really are only beginning to get an inkling of and the role that the microbiome is involved in. I think approaching it from infection control where it's a short, we understand the outcome we should get will really give us the tremendous insight that we need to understanding the importance of tending the microbiome. We have a question from the audience.
Jeff Fox, Microbe. I'm fascinated that you say 6 anaerobes seem to be fully adequate to restore the gut, whereas the gut's a mix of anaerobes and aerobes and all kinds of other creatures. How is that working? What do you think is going on?
Okay.
What takes care of the rest of that population?
Sure. Okay, so I should say 3 strict anaerobes, 3 facultative anaerobes, so it's a mixture of the 2. As I say, when we looked in these mice, when you look with enough depth, the other anaerobes are there. They're just very, very suppressed. And so in this case, what happens, like I say, is just that these 6 strains seem to be attacking the C. difficile. In some way, we haven't worked out the mechanism yet. We know that certainly it requires them to be live. It doesn't work with dead cells. It doesn't work with sort of the metabolic products, so the cellular filtrates from these cells don't work. Things like the short-chain fatty acids that they produce, they didn't have the same effect. So it seems to be an active living competition from these 6 bugs that does the job. And that allows the suppressed microbiota to come back and basically reestablish itself. I mean, I have to say, this does bring the question that the 6 bugs themselves obviously aren't enough to restore diversity. You need things to come back. And so it may be a case that in some C. difficile patients, their gut microbiota is so decimated by months on antibiotics, there simply may not be anything there to come back. And in those patients, you might still have to stick with the fecal transplant approach. However, in other patients where there's less perturbation and they're just suppressed, this simple mix is the key to unlocking that, I think.
Another question from the audience.
Pam Harrison, Medical Education Network. Dr. McDonald, could you elaborate on, I think you were suggesting that antibiotic resistance is really the result of pressure on the microbiome? Did I understand that correctly?
Well, antibiotic pressure, when we say that word, we don't really unpack it very well. We just say it and we think we all know what we mean by it. And certainly one aspect to it is from really classical understanding of resistance where you can apply an antibiotic to a single species over time and see resistant strains, mutants often, if there's no other genetic exchange coming into that strain, then it's mutant. If you have them mixed, then sometimes it is an exchange of information. But that's been our focus within antibiotic pressure on a single species, pressuring out the emergence and amplifying, well, not amplifying, but selecting, I should say, selecting, not pressuring, selecting the emergence of resistance within a single species. I'm saying, and that's been our that's where we've gotten most of our understanding of the emergence of resistance, but I'm saying that those events happen often without us being even aware of them. When we become aware of the resistance problem, is more often this transmission that has started between human hosts and sometimes animal hosts too. That is really afforded through pressure on the microbiome. Yeah. And that's, so again, from a public health standpoint, I'm speaking from that, the effect of antibiotics that is most important probably is its pressure on the microbiome. It's not that, the first emergence, classic one was vancomycin resistance in enterococci. We had vancomycin for 30 years. We'd seen no vancomycin resistance. Of course, we can all imagine that the genes, and there's a lot of genes involved in vancomycin resistance, they probably did come over from the Streptomyces somewhere where vancomycin was first, found, but it had to work its way over somehow. There are a lot of different genetic information and determinants to work their way. That was very important. I mean, when that first happened, that was important. I'm not saying that's not important. That's an important sentinel event for public health. But thereafter, the much more important factor is the effect on the microbiome. And this is where really I sort of think, as the CDC, and this is my personal view, this is not the official view of CDC. The 1996 guideline for prevention of vancomycin resistance was all about don't use vancomycin. Mm-hmm. In fact, one of those is use metronidazole to treat C. difficile, avoid oral vancomycin. Well, and a lot of people have commented since then that avoiding vancomycin doesn't do that much for your VRE problem once you have VRE in your hospital. Mm. Cephalosporins are driving it mostly. In fact, you'll see that. You can show that you give someone a cephalosporin, enterococci just go through the roof in their intestines.
It goes back to your tending the microbiome.
Exactly. And so, so that's and that's always been a problem when people have tried to do correlations with you're using this much of drug X and you see this much of resistance to drug X. Well, once the resistance has emerged in that particular species to drug X, it's drug Y and Z and all these other drugs.
Which is why I think your session today is going to be paradigm shifting, because it's going to really begin to help our clinical colleagues who are here at this meeting get a new perspective on antibiotic stewardship, because they're effectively looking at how many scripts they write and what the antibiotic resistant or their antibiogram of their hospital is. They're effectively trying to make the correlation, when in reality, I think the best lesson learned is the vanc cephalosporin story, because it really teaches a valuable lesson, which is why I think what we're going to begin to see is this paradigm shift of looking at the microbiome as a simple indicator for infection control in antibiotic resistance, and really to begin to help us to understand the antibiogram and whether or not stewardship is working.
Sure. And I'll say at CDC again, we've been guilty of that too. Know your antibiogram and prescribe according to, well, that's appropriate from the sense of you got a lot of this resistance, avoid that drug because you want to cover your patient. That's good. That's
patient safety.
That's patient safety. But in terms of looking at your antibiogram and saying, oh, all those R's are the drugs I should avoid using, that's not necessarily the true. I mean that might help as overall antibiotic prescribing goes down. But you still may have a lot of transmission of a certain type, resistant phenotype, resistant strain, even though you don't use any more of the drug that
Yeah.
you're
No, I mean, you see
labeling
this with
that.
C. difficile, right?
Yeah.
They go in and they've picked it up usually because they've been given clindamycin or something
Yeah.
that's broad spectrum. But then the patient's cured, they come back and they're treated for something
With
like a
anything
UTI
else.
or something with a very mild antibiotic,
Yeah,
and the C.
yeah.
difficile comes back. It's because
Right.
you've wiped out the microbiota.
Right,
So it's not that you're affecting the bug, you're affecting
Right,
the microbiota.
right.
So I mean, your point.
Yeah, and I think, and so I think this is a coming together of a lot of things. It's the attention to C. diff, which isn't by accident. I mean, I think the fact that we're seeing all this community-associated C. diff, we got to wonder also if the microbiomes of just general people in the community are being affected. You know, now 30% of the C. difficile that occurs with its onset in the community is in people who have not had an antibiotic in the last 3 months.
Well, is that because it's coming in from our food supply?
Well, that's a thought, but we're also finding drugs like proton pump inhibitors
Yes.
are associated with it. Probably, you know, I don't believe that's because of stomach acid suppression. These are spores that are highly stomach acid resistant. I believe it's microbiome disruption. That's why we need this index that could start to say, it's not just antibiotics, proton pump inhibitors, what do they do to the microbiome? Antidepressants, recently associated with C. difficile. There's a little bit about depression too, but I
then
mean, so.
some of the lessons learned from Dr. Walker's study, I was going to ask you, can we have you gained enough insight to move away from an animal model as your readout to using some of the molecular tools, transcriptomics, metabolomics, any of the omics that we now have in our armamentarium? Can you use those to effectively help Dr. McDonald get to his index?
I think that's the big question, isn't it? I mean,
Yeah.
we've said it a number of times. We're right at the beginning here. It's a very complex ecosystem, hundreds of different species. It varies. Every individual person has their own microbiota, which makes it even more complex. What's in your gut is different from my gut from your gut. So there isn't. I suspect there won't be a catch-all. This will work in every single case. I think we're going to have to be smarter. I think we're going to have to think about personalized medicine again in this sort of context. But certainly the new tools are absolutely going to drive this field forward. I mean,
Jeff Fox again. I want to go back to your comment about the vancomycin recommendation from 1996, because I don't see, I can't make the leap with you yet as to how information about the microbiome is going to give you a better recommendation at the moment that you can implement to spare vancomycin. Is there something else you can do that will be useful? Even though it's gradually eroding, as you point out, its susceptibility to vancomycin is being lost. What else can you do to try and guard against that?
Well,
At the moment, is there something practical?
I think that, well, at the moment is, I think the goal, the nearest term goal, I think is what you're also asking, would be if you could take a snapshot of someone's microbiome and just say, from this, they are at risk for VRE. Or maybe they even have VRE in that microbiome. And they are dominated by VRE. In fact, I'm going to throw this out, is that we could even, probably right now, give a non-absorbable that knocks down the VRE in that person, and then give them back a microbiome. I think that that's pretty close. I think we could do it with a fecal transplant. Certainly, I'm not saying that we want to do that necessarily because of other issues, but the problem of just saying vancomycin is the answer when it comes to antibiotics. It's clearly this person maybe had never had vancomycin, and yet they have VRE. How did they get it? Well, because they got cephalosporins, and they were around other people with VRE. So I don't know if that answered your question. Can you spare vancomycin? Also perturbs the microbiome also. But it was sort of the fact that I guess this is a question I'll ask you back. Not ask you back, but just say this: is that I don't think we know right now if you have VRE. Let's say you put a person in a ward with 30 patients and 5 of them have VRE. Do we know right now giving a 5-day course of vancomycin would be riskier for them than a 5-day course of cephalosporin for acquiring VRE? I don't think we know that.
That study hasn't been done.
Right, that study hasn't been done. It hasn't even been done in, well, maybe Lou Rice has done that, he might, but the point with mice, but the point is that the guideline would have suggested, oh, vancomycin in that situation would have been much riskier, okay? And I don't think that that was necessarily true.
Like I said earlier, it's paradigm shifting, and I think that's what's so provocative, and it's why this conference is so cutting edge, is because we're blending the different disciplines,
yes,
from the prescribers to the basic scientists to the public health folks, and
yeah,
all three of us have to get together to really crack this.
Well, I think it's an exciting time for microbiology, and I think I would really want to say again, pushing towards the infection control area because of the near term, that you have microbiologists in hospitals, you have infection preventionists and infection control experts. They both need to dialogue about this and start to understand it together as the tools become available. Now, I mean, everything I'm talking about right now is still in development. We've said it's early, early, but we need to start giving it some vision and direction so that people know it's not that far off. Anyway, so I think
This discipline is less than 5 years old. And with that, I think we're going to stop. You've been watching ASM Live at ICAAC, and we've been visiting with Cliff McDonald from the Centers for Disease Control and Prevention, and Dr. Alan Walker of the Wellcome Trust and the Sanger Institute from the United Kingdom. And you can learn more about this by attending their session this afternoon entitled Disrupting the Microbiome Through Antimicrobials, Antimicrobial Use, Implications for Infection Control. Also, be with us later today when we do This Week in Microbiology Live with Vince Racaniello, me, and a few other guests who are going to stop by and join us. So thank you, gentlemen. It's been a great pleasure talking with you this morning, and I think it portends to be a really exciting session this afternoon. Thank you.
Thank you.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.